स्टुडंट. नवयुवक शिक्षण प्रसारक मंडळ चापोली संजीवनी महाविद्यालय चापोली welcome you in e-learning platform prepared by myself, Dr. Shubhangi Chavan, Assistant Professor and Head Department of Botany. Now, Swami Ramanand Tirth Marathwada University Nanded Semester Pattern Curriculum under Choice Based Credit System CBCS for Undergraduate Program, Faculty of Science and Technology, Subject Botany, Year BSc Second Year. Now start Theory Paper 6 Plant Anatomy. Continue the unit Permanent Tissue. Last period, now see simple tissue and start the next tissue, Collenchyma. Collenchyma is a typical supporting tissue of growing organ in stem, leaves, floral part of fruits. It is also found in root, particularly the earlier roots. It is a living tissue composed of elongated cells, thick primary non-lipid walls. Parenchyma cells vary in size, shape. The smaller cells are premastic while the elongated cells are oblate or tapering ends. These cells are closely packed, may or may not have intercellular space. Collenchyma found beneath the epidermis or below outer layers of parenchyma. In stem, it is complete, slender, or in longitudinal strips, while in leaves, it is present in one or both sides of veins and along margin of the leaf blade. The cell wall consists of cellulose, pectic, and have high water content. Now, this is the diagram of Collenchyma. In first figure, Collenchyma in transverse section showing wall thickening. Figure show cell wall thickening, protoplasm and vacuole. Now, while in figure second, TS of and LS of Collenchyma cells are distributed in this figure, we can clearly see vacuole, extraangular thickening, cellulose wall, nucleus and cytoplasm. The diagrammatic representation of Collenchyma. Now, there are three main types of Collenchyma depending the basis of the distribution of wall thickening. First of all, lamellar, lacunar and angular. Firstly, lamellar. In lamellar Collenchyma, by the deposition of heavy thickening in the traditional than in the radial cell wall. Example for stem of Raphanus and Symbacus. Now, second, lacular. In lacunar Collenchyma, in which the thickening are primarily deposited around the intercellular space, Example, earlier root of monostera. And lastly, angular. In this type, the thickening are primarily at the corner or angular of cells. Example, stem of dhatura and lycopersicon. At the maturity, angular collenchyma develops in the annular collenchyma due to the thickening of the cell wall.